Hi, this is Genetics Lecture 3 discussing Punnett squares. Alright, so first of all, a Punnett square is a chart that shows all possible combinations of alleles that can result from a genetic cross. So down here in the corner with all the G's, you see what a Punnett square looks like. This is a completed Punnett square. Across the top here, I have my first parent, parent number 1. Down the side here, I have parent number 2. It does not matter in which order the parents go, just as long as the entire first parent is up here and the entire second parent is down off to this side. So, once I get my parents up there, I can cross their information and find out what their possible offsprings might look like. So, I'm going to take this G and I'm going to bring it down. Right there. Right there. I'm going to take this G and bring it down. Right there, right there. Then I'm going to take the big G and I'm going to pull it over. Okay? Again, right here and right here. Take my little G and I pull him over. Right here and right here. So, these are the possible combinations that the offspring of parents with the genetic crosses listed on the top and the side. So their offspring could possibly be big G, big G, big G, little g, big G, little g, or little g, little g. I think this will make more sense to you as we look at some more examples. All right, so blue is dominant over green. That means I'm going to assign blue the capital B, and the lowercase b will be assigned to green because green is recessive. My first parent here has the genotype big B, and the phenotype is blue. Okay. My second parent here is little b, with a phenotype of green. So let's draw a Punnett square to show this cross. First thing I'm going to do is I'm going to take my first parent. Hopefully you wrote it down on your paper. My first parent was big B, big B. My second parent was little b, little b. Okay? And like I said before, these parents are interchangeable. I could have put this one could have gone down here, and this one could have gone up here. It doesn't matter. Okay? What I am going to do is pull my letters down. So I need to pull my big B's down, pull them in all the squares, and I need to pull my little b's over, okay? Pull my little b's over. So in this combination, or this cross, this genetic cross, all of my offspring would turn out exactly the same. They are all big B, little b. All big B, little b. So, when you're doing Punnett squares, a lot of times you're going to be asked for a genotype, or you might be asked for a phenotype. Now remember what I told you in the last lecture, that when you're asked for a genotype, you need to give letters, and when you're asked for a phenotype, it should be words. So, we're going to represent these probabilities as a percentage, okay? Our total percentage should always add up to 100%, and each one of these squares represents 25% chance. So in this case, if I have 25 plus 25 plus 25 plus 25, I'm going to end up with 100%. So these children from this cross will be 100% big B, little b, no questions asked. Now, what are my phenotypes? Well, remember we said blue was dominant to green, so all of these children carry that big blue allele, which means they are all going to have blue eyes. So we would say blue 100%, okay? And that is basically how you do a Punnett square. We're going to try another one just kind of like this one, just a little differently so you get the same idea. Here we go. In this example, we have blue is dominant over green. Again, big B equals blue, little b equals green. In the first parent, this time, we're going to have a big B, little b, or a heterozygous parent, all right? And their phenotype is still blue. Down here, my second parent, again, big B, little b, heterozygous parent, and their phenotype is blue. So let's try this cross. Here we go. All right, again, we put one parent across the top, big B, little b. Put our second parent across the side, big B, little b. Now, I'm going to perform my cross. I'm going to bring my big B down. I'm going to bring my little b down. Bring this big B across. I'm going to bring this little b across. All right, so in this example, my offspring are not all the same. I have some differences. So I'm going to have to show that when I do my probability in my genotype and my phenotype. So let's look at our genotypic ratios first. We'll write down our three possibilities are big B, big B, big B, little b, and little b, little b. I have all of those genetic crosses in these squares. These are possible outcomes. I look at this like it's a dollar bill. I've got 4 quarters of a dollar bill, and those are my percentages. So, how many quarters do I have for big B, big B? Just 1 quarter, right? So that means he's going to be a 25% chance. Now, how many quarters do I have for my big B, little b? I have 2. 2 quarters equals 50 cents, or I have a 50% chance of having big B, little b in these offspring. Now my final one is right here, little b, and he's only, what percentage chance? We already have 25, 50, that leaves me with 25%. Okay, those are my genotypic ratios for this cross. Now, let's talk about our phenotypic ratios. They're a little different. My phenotypes here. Now, again, treat them like they're all 4 quarters. They're going to all add up to 100%, okay? So, remember that if I have a dominant B, I'm going to have blue eyes, right? So in this cross, I have 1, 2, 3 of the quarters are all going to be blue eyes. So if we add that up, that is a 75% chance these 2 parents will have a blue-eyed child. Now, we have this 1 quarter over here that is green because there is no dominant blue. So they also have a 25% chance of having a green-eyed child. Take a few minutes and think about it. Go back and watch it again if you need to. We're going to write down some steps now to kind of help clear things up even more. So this is kind of what we just went through, is the probability of the offsprings, the genotypes, and the phenotypes. We can look at the probability that each of those is going to happen. So for this cross, we're gonna break it down even more into steps. So in this one we have purple people eaters. 1 horn is dominant and no horns is recessive. We're gonna draw a Punnett square showing the cross of a purple people eater that is hybrid for horns with a purple people eater that does not have horns. What are the possible genotypes for this cross? Now, there's a couple things when you read word problems like this, and a lot of your Punnett square problems will be word problems. You have to underline or you have to highlight in some way the important parts. And the first thing I always look for is what is dominant to what. And in this case, it says 1 horn is dominant, no horns is recessive. So automatically, I'm thinking about assigning letters to these guys. I'm going to say the big H is for one horn, because that is dominant, to a little h, which is no horns. Now, the next thing I'm really going to look for is what my parents that I'm crossing, what are they? All right? My first parent right here says he is hybrid for horns. Hybrid means the same as heterozygous, which means he has two different alleles. So hybrid for horns should automatically translate in your brain to a big H, little h. A big H, little h. Now, let's look and find our second parent. Our second parent here, it says that this guy or this girl does not have horns, okay? No horns. And since no horns is recessive, the only option here is that this parent is a little h, okay? Now, the final thing I need to look at is what is the question asking me for, because this will trip you up a lot. In this particular instance, they're asking for genotypes. Genotypes, which means my answer needs to be in letters, not words, letters. All right, so let's move on here. So step one is to assign letters or alleles to the traits you're testing, and we just did that. We said that big H equals one horn and little h equals no horns. Okay, that was our first step. Step is to set up the Punnett square, put the first parent across the top and the second parent across the side. So if you draw your Punnett square, across the top we have our hybrid or heterozygous parent. Across the side we have a homozygous parent with no horns. So there are my parents. Now in step 3, I'm going to perform my cross. I'm just going to write my parents back in here real quick. And now you want to do your cross. You want to pull down your H's. Big H, pull down the little h. I'm going to pull this little h over. Pull this little h over. All right. So here are my offspring. Now, step 4 is to evaluate your probabilities. And we'll go ahead and do that right here. We want to check the question and make sure we know what it's asking for. And if we remember, it was asking for the genotypes, which means I need to give letters. All right, so I have two possible outcomes here. I have a little h and a little h. Okay, so let's write that one down, h. My other option here is a big H, little h, and a big H, little h. So I'm going to write that down, big H, little h. Now, what percentage are each of these? Well, the little h is going to be 2/4 or 50%. And the big H, little h, again, 2/4 or 50%. So that is how you do a Punnett square. I hope this was helpful to you. Let me know in class if you have any questions or you would like me to go over these further.